In this video, we take on transcription, the relatively simple process, habits inside the nucleus, getting us from DNA to messenger RNA using complementary base pairing. Going over the details of transcription, I'm going to break it down into these three phases, and they're shown on this diagram over here: initiation, elongation, and termination. Don't make too much of these. Really what this means is that we need to start somewhere, we need to keep going, and then we eventually need to stop. Can't just keep going forever. So, not very complicated. But the most difficult part, really, when you think about it, is initiation. This is where our enzyme, RNA polymerase, finds out where to start. Remember, there's about 2 meters of DNA in each human cell. Those are divided into 46 chromosomes, and those contain 22,000 genes. And out of that, we want to find one. So RNA polymerase, it can transcribe any gene, but it's got to find the right one. So this gene here, we've illustrated these two strips here as double-stranded DNA, and the gene just basically goes from here to over here. But for the RNA polymerase to find the right spot on the gene, it needs a promoter. It needs some help finding that specific gene. You might think of it like this. The chromosomes in a nucleus are a lot like these bundles of yarn in a basket. And in order to find the place to start, it'd be nice to have a sign that says, "Start here." So now that initiation is done, we move on to the elongation step, where we're just going to continue the work that was started here in laying down complementary bases and making a longer and longer piece of messenger RNA in the pink, although I will note that the same exact process can be used to make any type of RNA, which is transfer RNA, ribosomal RNA, and even others. Remember, that process is driven by complementary base pairs, so if the blue strand is DNA and this right down here in brown is our RNA, then complementary base pairing, A on the DNA strand codes for U on the RNA strand, T codes for A, G codes for C, and C for G. Basically the same as DNA, except for this uracil, which takes the place of thiamine when it comes to the RNA. So that's elongation. The last step, but certainly not the least, termination. We need to know, where do we stop? The gene can't go on forever, and this is not DNA replication. We're not turning the entire chromosome into a piece of RNA, but we need to reach this termination site, right? A piece of DNA that says, this is it, you have reached the end. And essentially what happens is your DNA goes back together. So this little bubble that was here closes up. DNA is safe and sound, stays in the nucleus. Our messenger RNA transcript is released, and so is the RNA polymerase, ready to find a new gene and do its job again. If you're up for it, let's take a look at transcription again using the DNA paper models from the class. So we're in the nucleus. We have double-stranded DNA, 2 meters of it in fact, but we just need a gene. There are lots of RNA nucleotides floating around and an RNA polymerase in search of a gene to transcribe. Now, you may not think it's very difficult for RNA polymerase to find the gene, but it's more difficult than you might think. All right, so this promoter part of the gene is important. It's a strip of DNA that attracts other proteins that tells RNA polymerase, land here. Now, very much like its cousin, DNA polymerase, RNA polymerase has the same rules. It goes from 3' to 5', laying down complementary bases. It also splits DNA as we go. This is the coding strand, so this is the non-coding strand. We only need to use one piece. And so the rest of it
works very similar to DNA polymerase. We're going to add complementary bases starting right after the promoter. Now as we come to the end, we reach the termination site, and that's how the RNA polymerase knows it's reached the end of the gene. Normally this would be several hundred nucleotides long, but we just don't have time for that. So we're going to place that. RNA polymerase is going to fall off. So this is what's known as termination, obviously. This can separate. Covalent bonds are formed between all the nucleotides here, and we have our messenger RNA. This, by the way, is the same way that other types of RNA are made, including ribosomal RNA and transfer RNA. But in this case, this represents our messenger RNA.